Liz.
Okay, I'm going to take Roy seriously and sit at my seat and give my talk. I hope you can see me. So I'm Liz Mansfield. I'm the director of the personalized medicine staff, and I'm actually in the Center for Devices, but have been working on the policy around companion diagnostics, which is actually sort of a drug issue. But Rick and I get along pretty well. Very well. Who am I pointing at here? Okay. So you're probably all aware of the policy that's sort of been floating around out there since about 2008, and that is that FDA wants approval of a test if it's essential for the safe and effective use of a therapeutic product. And this has come into play in the vast majority of cases in oncology, partly I think because oncology is a genetic disease, and partly because therapeutic development is much more rapid there, and things get approved earlier in smaller trials. So we formally articulated the policy in 2011. We have about 11 different drug indications that have fallen under this, and it seems to be a successful approach. But I can tell you right now, we're using a sledgehammer where, in fact, we'd probably like to be using something much smaller and finer to get at cancer, as many people in the previous panel have alluded to. In vitro diagnostics, where most of the companion diagnostics lay right now, is an area of super rapid evolution right now. You all know next-gen sequencing. It's practically not new anymore. Digital PCR, circulating tumor cells, proteomics, you name it. We're capable now of doing multiple simultaneous measurements on one sample. So in fact, the idea of having one test per drug doesn't make a tremendous amount of sense for the future. Right now, we're mostly nucleic acid based, so until somebody brings forward technologies that look solid somewhere else, we'll stay there. And the model of multiple simultaneous measurement is already in use by what I call the early adopters, primarily the academic medical institutions and some biotech development companies. But I want to assure you that we're tracking the technology, and in fact, we're very interested in some of this new stuff. And I would put out a cautionary note that because nobody has done it yet doesn't mean that FDA won't allow it. So I've heard that too many times, and I don't want people to walk away with that impression. I don't know what I'm pointing at, so there we go. So the changes in policy that I envision coming forward, that need to be well informed, or the ability to use these multiplex technologies, knowledge engines, and so on, given a particular cancer type, given any kind of cancer, where we can measure all or most of the markers of interest so that we get many results from one sample. And we need to understand exactly what the appropriate policy is moving forward to transition some of the older tests into this, and what we're going to do about tests that don't fit into this paradigm. And the questions that need answers, I guess I pose this a little bit differently, are what do we need to know about the test? There are lots of unknowns out there, whereas we're talking on a very high level before about systems biology. We still don't understand tumor heterogeneity in terms of response. We don't understand the test sensitivity in terms of probable response. If you have 1% tumor cells that have a mutation, will that tumor be expected to respond? And especially, I think this has been a big area for us, is primary versus metastatic disease. We tend to see patients coming in, they may have metastatic disease because they're in early stages of drug development, but the tumor that was tested for the marker was actually the primary tumor, because the metastatic tumor is either inaccessible or the patient is not up for a biopsy. So there's a lot about biology we still don't know, and I think making the quantum leap to systems biology would be great, but we still have some of the very pedestrian questions laying around. So what do we need to know about the biology as well, is in terms of mutations in DNA or changes in protein, epigenetics, whatever. What are the spectrum of mutations or changes versus response that we ought to be thinking about? In fact, we have no idea right now. To some degree, we've been limited by technology that you can only measure a certain number of things. So you can't see the whole spectrum. You can only see what you focus your microscope on. we're running more and more into, as we subclassify disease by molecular analysis, that we're seeing more rare or private or previously unseen mutations coming along, and we don't really know what to do with those, and we don't know what they mean in terms of response. So these variants of unknown significance or new pathways, we don't exactly know how to match those up to a therapeutic option. So we would actually are looking for ways to try to answer these questions. I just keep pressing it. And finally, well, I wanted to float, it's not on my slides because it's so cutting edge that I just thought of it. No. One of the things that I think that I'm interested in getting response back in the policy area is, in my part of the world, is in vitro diagnostic tests, and we also look at radiology and so on, is right now we're clearing and approving tests that are tied to a particular therapeutic action. In fact, in cancer especially, many cancers are, as you well know, deadly. People don't have long. And in many cases, there's no well-known treatment for them. And one of the questions I have is, would it be useful for FDA to authorize a test that would actually be primarily for use in sending patients to clinical trials, to send them to therapies that we don't know if they work yet when they've exhausted other therapies. So that would be a really new paradigm for us. It would be sending patients into the unknown. And would love to have reaction on what the value of that would be, and sort of the pros and the cons. You can think of a lot of cons of people thinking, using things inappropriately. And I also worry a little bit about how systems biology is going to be accepted by the community oncologists. I don't even understand the systems biology. I'm not sure that every community oncologist is going to be able to unravel all of that. So while I really hope it happens, I think there's a lot of learning that needs to happen in between. So let me end up with saying, kind of quoting somebody who you might have heard of before, and probably loosely quoting is we need to figure out what we know we don't know, what we don't know we don't know. And it's time to start asking questions so that we can get more answers, even on the fundamental side, especially to underpin the systems side. Thanks.
Great, thank you, Dr. Mansfield. Are there any questions now, or we can certainly wait till we have the panel and let Rick speak? Okay, seeing none, then we'll move right to Rick, who I'm sure is well known to most of the group. He's the Director of Oncology Drug Products, and now the Director of the Office of Oncology Drug Products at the FDA. He's had a long, distinguished career in academia and at the FDA, and he's going to give us some of his perspectives on these issues. Rick.